So in today's video, we're going to be talking about the phospholipid bilayer that makes up the plasma membrane of cells. So you'll recall that phospholipids are composed of a polar head region and these nonpolar tails. And it's energetically favorable for phospholipids to form a bilayer in an aqueous environment. That's because the polar heads are hydrophilic and they like to interact with water, and the tails are hydrophobic. They're made of hydrocarbons, and it's energetically favorable to sequester those tails away from water. And so these phospholipids naturally form a bilayer. And depending on the domain, you can end up with different hydrophobic tails. And so in bacteria and eukarya, so in our cells and in bacterial cells, these tails are fatty acids, so hydrocarbons that are just really basically a long stretch. And in archaea, they're isoprenoids, so they're these hydrocarbons, but then they have these methyl branch groups coming off of them. And this gives these phospholipids a bit more rigidity. Okay. Now, there are other things in the plasma membrane. We have in animal cells, we have cholesterol, which is a steroid, right? Cholesterol has that hydroxyl group and then that characteristic 4-ring structure followed by an isoprenoid that's attached to the final ring. But in plant cells and other species, you can have steroid, or in other domains, you can have steroid-like molecules in the plasma membrane. The plasma membrane, or the phospholipid bilayer in cells, is what's called a mosaic. So not only does it have lipids, but it also contains proteins. And this green tube here represents a protein that's embedded in the membrane. And there's two types of protein that can be associated with the membrane. We can have integral membrane proteins, which are embedded in the membrane, or we can have peripheral membrane proteins, which are basically just attached to the membrane. And the phospholipid bilayer is fluid, meaning that lipids can move in the bilayer. And so here's a lipid moving over time in the bilayer. Okay, so the fluidity of the membrane, right, how fast these lipids are moving, influences its permeability. So let's talk about permeability. Okay, so this membrane here that is separating the outside environment from the inside of the cell is selectively permeable. Okay, so small nonpolar molecules can move quickly across the plasma membrane, but charged or large polar molecules either can't cross or go through very slowly. And so we're going to walk through these examples. So here is oxygen, right? I'm going to grab my pen. And because oxygen, even though it's electronegative, right, compared to other atoms, compared to itself, it has the same electronegativity. So these electrons are shared evenly, which makes this double bond a nonpolar covalent bond, okay? And then CO2, right, the electrons are not shared evenly. They're closer to the oxygen compared to the carbon. But because this molecule is symmetric, right, this whole molecule is not nonpolar. Okay, so let's go back to the clicker. Maybe if I just hold still. So these will rapidly cross through the membrane. So if you want to see that again, these are small nonpolar molecules and they readily cross the cell membrane. Okay. Water is a small polar molecule. Okay. So it's polar, which means it's not really going to like to interact with the hydrophobic tails and the lipids. So that's going to slow it down a little bit, but it's still going to be able to pass through this semi-permeable membrane. So it's going to go slower than those small nonpolar molecules, but it's still able to pass through. Glucose, right, is somewhat a medium-sized polar molecule, right, because there's an oxygen here and there's some hydroxyl groups in the glucose. Okay, now again, right, the polar parts don't really want to interact with the hydrophobic region, so this is going to slow its movement down through the membrane. Okay, so it's going to kind of bounce and then maybe eventually go through. So it goes through, glucose will go through at a slower rate. And then the last example are ions. So these are charged substances or charged molecules. And these charged molecules really like to interact with water, but they do not like to interact with the hydrophobic tails. And so really, these ions are basically not permeable without some sort of channel, which we'll talk about in the next class, to get them into the cell. And we basically learned all this by using these experimental systems to discover what would go through a membrane, a cell membrane. And these experimental systems are called planar bilayers. And what they do is they take, here's a flask that has this gap in between them. These are usually solid plates. And then they allow a lipid bilayer to form between this gap. Okay, so here, if you blow it up, here's your lipid bilayer with the tails facing each other and the polar head groups facing the aqueous environment. And they put a solute or an ion on one side of it, and then they wait and measure if it goes through that semi-permeable membrane. Okay, so again, if we were doing this and this was a cell membrane, we would see that small nonpolar molecules would pass through rapidly, followed by small polar molecules, followed by medium-sized polar molecules, and then ions basically wouldn't pass through at all. Okay, and that's again summarized once more. So we have our small nonpolar molecules that pass through really fast, and then water passing through medium, glycerol, maybe sometimes going through, other times kind of bouncing off. Glucose, doing that, going through, bouncing off, and then the ions really not being able to pass through the semi-permeable membrane. And so a hint for the daily quiz, I want you to be able to know what would go through quicker, something like oxygen or something like an ion. So this is a helpful chart to take a look at before the quiz. So we've talked about how membranes are semi-permeable and the types of molecules that go through really fast and types of molecules that don't go through at all. But there's features within the membrane that allow it to be more or less permeable, okay? And really what that means is that the membrane is either more fluid and more permeable or less fluid and less permeable. So what factors influence fluidity? Okay, so always keep in mind, again, more fluid means more permeable, so things are going to go through more rapidly if the lipids are moving a lot in this membrane. And less fluid means less permeable, so they're going to pack in really tight and not allow molecules to go through. And the things that affect membrane permeability are the number of double bonds in the hydrocarbon tails, the length of the lipid tails, the number of cholesterol molecules in a membrane. Remember, cholesterol is just in animal cells, but other steroids too, and temperature. We're going to talk about each one of these variables in a little more detail. So let's just review bond saturation. So here is an example of a fatty acid, right? And this fatty acid is saturated, which means that there are no double bonds in this fatty acid, right? Because each carbon is saturated with hydrogens. Okay, so this is a straight tail, or a straight tail. And then here is an unsaturated fatty acid in which one of the carbons is double bonded to each other. And what this does is it creates a kink in the fatty acid. Okay, so this one's going to be bended a little bit. And if you put this into a membrane, a phospholipid bilayer, what happens is that if you have all the lipid tails are saturated, so they're straight, there's fewer spaces and stronger hydrophobic interactions between them. And so the membranes are denser and less permeable. So these, if you have straight tails, they're saturated hydrocarbons in those tails, the membranes are less permeable. If the membranes have kinks in them, or the tails have kinks in them, meaning that they have double bonds or they're unsaturated fatty acids, this prevents that close packing and it reduces the hydrophobic interactions. And so molecules is much more able to get through and the membranes are more permeable. So disclaimer, the wiggly lines that I've been using throughout don't usually represent anything about saturation. I only used it for this example. Okay, so just to review, saturated here, meaning no kinks, they're able to really pack it in, they're less fluid. And then this example here, the unsaturated allows for kinks, meaning that they can't pack in and they're more fluid. So here's glucose, taking its time going through that one. And then here's glucose moving through this membrane much more quickly because it has unsaturated fatty acids. Okay, so saturated, no kinks, less fluid, less permeable. And unsaturated, meaning that it has kinks, so it can't pack in, meaning this should say more fluid. And more fluid means more permeable. Okay, so that's one, bond saturation. The second one variable we can talk about is tail length, okay? So in this example here, the tails are really, really long, okay? So they have much more carbons in these tails. Maybe they've got 20 carbons in the fatty acid tails, and here maybe these ones only have 14, okay? So longer tails, meaning more hydrophobic interactions, means that this membrane is less fluid and less permeable. Okay, those hydrophobic interactions become stronger as the saturated hydrocarbon tails increase in length. Okay, and those shorter tails are more permeable. Okay, because they're able to get in, they're not packed in as tight. So to show you, here we have glucose coming in, taking a really long time to get through that membrane, and then here going through those shorter tails much more quickly. So less fluid, less permeable if the tails are longer, more fluid, more permeable if the tails are short. And this is just a summary here of those two features combined. Here we have kinked fatty acid tails that are short, and this leads to higher permeability. And here we have long saturated hydrocarbon tails, and this leads to less permeability and less fluidity. So the next variable I want to talk about is adding cholesterol to the membrane. And adding cholesterol to the membrane increases the density of hydrophobic regions, okay? So now we're adding even more hydrophobic regions to this region in the lipid bilayer, this hydrophobic region. So more hydrophobic interactions. And because of this, cholesterol decreases plasma membrane permeability, compared to this membrane here, which only has a little bit of cholesterol, so less hydrophobic interactions, or less density in the hydrophobic section. So this membrane here with a lot of cholesterol would be less fluid and less permeable, and this with a little bit of cholesterol, this membrane here, more fluid, more permeable. And finally, the last variable that influences membrane permeability is temperature. And so cold temperatures reduce membrane fluidity because they basically reduce movement. So phospholipids in the plasma membrane move laterally within the bilayer. Right? Remember that red phospholipid bumping around. And membrane fluidity decreases as the temperature drops because the molecules in the bilayer move more slowly. And when they move more slowly, those hydrophobic tails come together more tightly. Okay, so again, that denser hydrophobic region. And then decreased membrane fluidity causes decreased decreased permeability. So less fluid, less permeable at low temperatures, higher temperatures, more fluid, more permeable. So that's the high temperature. So in summary, less fluid membranes have saturated fatty acid tails, long fatty acid tails, high cholesterol, and low temperatures. And more fluid membranes have unsaturated fatty acid tails, short fatty acid tails, a low amount of cholesterol, and at higher temperatures are more fluid. And so that's the factors that influence membrane fluidity. And then in the next podcast, we're going to talk about how molecules get through via diffusion and how water crosses the membrane through osmosis.